What if we told you that in the not too distant future, you could log onto your computer and program living cells like software? Ever since scientists unlocked the secret language of biology, DNA, we've made massive strides in understanding the base code that underlies all living things. And over the past few decades, we've become so obsessed with this code that scientists are now exploring how to edit genomes and even create brand new ones that never existed before. So how close are we to harnessing synthetic life?
Life is a technology. It's a technology we didn't create.
But what if we could learn to harness it? That's the principle behind synthetic biology, the emerging study of building living systems. You might be imagining armies of Frankenstein creatures, and we'll get to that. But what we mean for the time being is creating animal products or individualized medical therapies, even transplantable organs, all starting with synthetic DNA and cells in a lab.
I look at them as 3D printers, but the most powerful and successful 3D printers that exist today, because they can print thousands and thousands of different materials all at once. And they can make more of themselves. And it comes with a programming language, DNA.
Pulling the principles of engineering design and then the real fundamentals of molecular biology together is really powerful, because it makes you think of biology as a design kind of medium, if you like, using the genetic material as the building blocks.
And when it comes to synthesizing life, there are two main schools of thought.
One is actually trying to build artificial cells. So actually starting from the bottom up and saying, how do I build a living cell in a kind of synthetic way? You're either using sort of synthetic chemical components or using natural biological components. How do I build a cell from first principles, from scratch, basically, bottom up?
In 2010, Craig Venter's research team made history with this approach when they created the world's first synthetic organism, a bacterium made entirely of synthetic DNA.
And most recently, out of the UK, Jason Chin and his team synthesized the E. coli genome. That's pretty remarkable. That's a 4 million base pair genome.
So we now can specify we want a genetic sequence, A, T, G, C, T, 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 A, whatever. And we can send that information across the web to a company, and then they will put it into their production pipeline, and then we will get sent back a chemical synthesized piece of DNA.
It's only going to get faster and cheaper and easier from here.
But it's still not as fast and cheap as editing an existing genome to synthesize new life. And to be honest, starting from scratch might not be worth it.
A purist would say you have to make all the cellular components from atoms. Okay, no cheating, no using bits of life. You can make fully synthetic DNA chemically, but for most purposes, it's not any more useful than something that's semi-synthetic.
Now, the other way is actually taking existing cells but re-engineering them to have particular functionality, to the extent of completely resynthesizing their whole genome and redesigning it, but putting it back into the cell, which means it's still a species, but it's got a human-designed genome in it. Building that code to perform a function or to do a design or to make something.
And that's just what we're beginning to explore. With genetic engineering tools becoming more accessible than ever before, researchers want to use synthesized genomes to enhance human health.
For simple things like detecting infections or detecting environmental pollutants, we can now engineer bacterial cells that will detect arsenic or fluorine or any of these toxic chemicals, and will change color like a pH strip.
These bacteria could protect us from consuming toxins in contaminated drinking water, for example. And Andrew and his team at Humane Genomics are working to develop a library of synthetic viruses that could lead to customized cancer therapies.
So the virus is like a USB stick. You know it's going to dock with the particular cancer cell. And it's going to load a program into that cell. Now we get to write that program to make it even more specific for the cancer cell. We're working with dogs and dog cancers to gain experience. The cancers that we're working with, first, it's a bone cancer. So we put in little switches into the viral genome so it's only active in a bone cancer. And you can measure its killing activity relative to normal cells. Like the first attempt at anything, it's not going to cure cancer. But we demonstrated that we could go from a file on a computer to a virus particle that could infect a cancer cell. And we did it in a relatively short period of time for relatively low cost.
Other researchers hope to take on the fashion industry with lab-made versions of materials like leather or silk. But perhaps the most ambitious plan involves bioremediation, preventing or counterbalancing ecological damage using microorganisms.
The concept is you have organisms that are engineered that would go out and either, start degrading plastic, start using plastic as an energy source, possibly start using toxic components, cleaning up environments using natural microbial systems.
Okay, we know what you're thinking. What about synthetic humans? Enter Human Genome Project Right, or just Genome Project Right. A sequel to the Human Genome Project, GP Right is bringing together over 100 institutions worldwide in one communal quest to synthesize the human genome.
You want a human cell which has new properties. You want to make something that has new properties like virus resistance or senescence, aging resistance. So you could do it with organic chemistry, or you could do it with biochemistry, or you could do it by editing of living cells. You could build human parts, meaning transplantable human parts, and from that you could build large portions of a human being. But right now, I think we're in a time where that's on hold, in part for lack of strong arguments for why you would want to do it.
Instead, these researchers are looking to understand the genetic symphony that makes humans tick by demystifying our cells' instruction sets.
The simplest microbe in the world, right, which has got the smallest number of instruction sets, we don't even know what 40% of those instructions do. Our knowledge base is still quite limited. So it's like redesigning an airplane with components, and half of the components you don't know how they function. So that's where we're at.
And if you think tinkering with genomes is a bad idea when we know so little about them, you're not alone. I mean, what if something goes wrong? Should I live in fear of evil synthetic humanoids taking over the world? Ah!
I think fear and concern and dystopic narratives is a good thing. I think it's far better to have too much discussion of how things can go wrong than having too little.
We've been using living organisms to produce and manufacture things for thousands of years. We've been using yeast to make bread, alcohol, but what we're doing now is much more systematic and just opens up all the possibilities.
We have to frame it as the exploration of inner space. Instead of going up into the stars, we're going down into the cells, learning how to get there, how to explore, and how to manipulate. It's as important as going out to the stars, because if we get to Mars and we don't have a way to grow food and to keep ourselves healthy, we're not going to survive there for very long. We just have to start opening the doors and saying, come on in, take a look. Don't be afraid. Let's all learn and grow and use this incredible technology for good.
So, if DNA is really the next HTML, how close are we to harnessing synthetic life?
This is a very fast-moving field, and I think, in 10 years' time, 15 years' time, we will know enough about microbial genetic engineering to be building microbes that could be potentially quite novel.
We went from 2015 being able to edit 2 genes to 2017 doing 25 genes in the germline, and now we can do 26,000, which is actually relatively easy in the era of exponentially improving technology. I think we could have mammalian systems, including human, that are multivirus resistant within, I'm guessing 2 to 6 years.
All you have to do is be able to write a new genome that nature hasn't created. And by that definition, I'd say we're already in the age of synthetic life.
We're quite far away from even thinking about a human, but as the technology increases, there'll be more interventions into multicellular organisms, and we need to be mindful of what they are and have good reasons for why we're doing it.
It's in your DNA to love science. So click the subscribe button and let us know down in the comments below or over on our community tab what big question you'd like us to tackle next. Thanks for watching.